Well, ladies and gentlemen, welcome to this lunch hour lecture. Thank you all very much for coming. I hope you'll enjoy the next hour. My name is Andrew Kopp. I'm professor at the Institute of Child Health and very pleased to also be a work colleague of our speaker, Dr. Gabriel Galea. Gabriel started his training as a veterinary surgeon. He did his degree at the Royal Veterinary College here in London and also at the University of Bristol. But then he took the most unusual step, I think, for vets of stepping out of his training for a few years to do a PhD in research. And that's when I first met him, 6 years ago now, when he was carrying out his research in the University of Bristol, mainly on, and he may mention this as when he speaks on bone development, because the research group he was in there was particularly interested in bones in relation to veterinary practice. And Gabriel became very experienced in biomechanics, the mechanical properties, of course, very important for bone and the skeleton in general. But we began to collaborate on some particular experiments of his, and we got to know each other, and I was very pleased then that after a couple more years, he was able to get a postdoctoral fellowship from the Wellcome Trust to come and join my group as a postdoctoral fellow. And so he was able to bring his expertise in biomechanics to our work, which for many years has been on birth defects, which occur during embryonic development, and particularly of the nervous system. And so the main defect that we work on, and Gabriel will talk about this, is spina bifida. And so Gabriel came, he did some fantastic work over the 3 years of his fellowship, and then quite recently applied back to the Wellcome Trust for a career development level fellowship, which is a fellowship that allows you then to become independent. And so now he is no longer a postdoctoral fellow in my group, but he's a colleague of mine running his own research group. So this has been a great sort of journey for Gabriel, moving from bone to the neural tube, as you'll see, for us, bringing in this superb new technology and Gabriel's know-how. But he hasn't left behind his veterinary roots. He's still, amazingly, despite the amount of work he does in science, also goes out on the streets as one of the leading members of Street Vet, which is an organization in many cities, I think in the UK now, that provides veterinary care to animals owned by homeless people. And again, maybe Gabriel would just mention that. So that's what he does in his spare time. So anyway, that's enough from me. Put up your first slide. So Gabriel's going to talk to us about his research. How do embryos build their spinal cord?
Thank you very much, Andy. And I'm actually really excited to be here today for a couple of reasons. First of all, the broad topic I'm going to be talking about, it has been in the news quite a lot recently, so I'm hoping lots of people in the audience will have heard about it, will be specifically interested in it. The second reason is because I'm mostly be using pictures that look a bit like that. What's happened over the past few years is we've developed this ability, thanks to brand new microscopy techniques, to take what used to be schematic diagrams of embryos in textbooks and turn them into something that looks quite pretty, I'd like to think. And that is actually the developing embryonic spinal cord. It doesn't look like a spinal cord, I know. So if we think of our spinal cord, it connects our brains to our muscles, our skin, and so on. It's quite a delicate structure. It's covered over by bone and muscle, and then ultimately the skin overlying everything. But it starts off in the embryo looking something like that. All right, let's get started. So, you may have seen articles like this in the press recently. These are news stories that describe a heroic surgery that can now be performed on fetuses while they're still inside the womb. So in this operation, which can only be performed in a small number of centers around the world, including here in London as of recently, the surgeons are actually able to go in and repair to an extent birth defects that have happened during embryonic development. And the main defect they're designed to help with is this one shown schematically here. This is spina bifida. So this is a birth defect, so a defect which will be present throughout the individual's life, in which the spinal cord at birth is not covered by bone and muscle and skin, but rather is exposed to the outside environment, and as a result, the cells, the neurons in it, start to undergo a degree of damage. And as a result, the individuals with spina bifida are going to have less ability to use their legs, to control their bowel and bladder and so on. And it does require surgery, at the very least at birth, to cover over this defect and keep it protected. It's not a rare condition. There are about 200 patients born with spina bifida in the UK every year. And that's despite the fact that there is a preventative strategy that is already available. So many people here who know about spina bifida will know that women who are expecting to become pregnant, in many cases, will take folic acid supplementation before and during gestation. Quick show of hands, how many people here have heard of folic acid? All right, good. That's a really good number to see. I mean, one of the concerns is that people might not be aware of the benefits of folic acid. So folic acid will reduce the number of cases. And like most things worth mentioning, folic acid supplementation is somehow impacted by Brexit. But it's not the be-all and end-all. Folic acid does not currently prevent all cases. What it does give us is a really good proof of principle. A proof of principle that a small molecule, in this case a vitamin metabolite, which women can take before and during pregnancy, can reduce the likelihood of this birth defect. Now, as Andy mentioned, I'm a vet by training, and I do still do a bit of vet work every now and then, mostly through street vet. And for the sake of this presentation, I asked my colleagues in practice to send me images of animals they have seen with this condition, with spina bifida. And I got inundated with everything from pigs to puppies with spina bifida and associated conditions. There is a puppy suckling from its mother up top in a rescue center in Thailand. And that raised red, raw lump of tissue on its back, shown by the arrow, is a spina bifida lesion. As Andy mentioned, my background's in bone, and here's the vertebral column from another animal. And the red bit I've indicated at the bottom is where the bone hasn't fused over, and you would have exposure of the neural tissue through the skin to the outside environment. Again, we can do x-rays and so on to get a better picture of what's going on, although in animals we don't tend to treat spina bifida. Unfortunately, in my patients, which is not the case in human patients, in my patients they don't tend to survive. This is my favorite. This is a lamb. This is a pet lamb. Pet lambs are incredibly rare. Pet lambs with spina bifida, I've probably got the only picture out there. But this is a pet lamb that has a spina bifida lesion right at the very end of its spinal cord. And again, if you compare these two images of two different mammals like us, which have spina bifida lesions that look exactly like they would in humans, but the mammal which has really taught us most about spinal cord formation and about the development of spina bifida is that powerhouse of biology research, the mouse embryo. Here's a mouse embryo right there. You know it's a mouse embryo because I've told you it's a mouse embryo. This is at a stage in development where even a vet struggles to tell species apart. This mouse embryo is minuscule. It's tiny. I've put it there next to a 5p coin, but I realize no one carries money on them anymore, so there it is next to a debit card chip. It's less than a third the size of a debit card chip. It's absolutely minuscule. So let's magnify it under a microscope. And now hopefully you can start recognizing some structures that look familiar to you. It's got an obvious head, an obvious body. And the body tapers round into what looks a bit like a tail, although it hasn't quite yet formed its tail yet. The head has the future brain in it, and the future brain is connected to the future spinal cord. And at these stages, the brain and spinal cord are one continuous tube. The same in us, postnatally, our brain is connected to our spinal cord as a single long structure. In these early developmental stages, it's really quite a simple tube, the so-called neural tube. We're not very creative with our naming, and this is a topic that will come up again and again. But what I'm showing you here in cyan and magenta is the neural tube, which is effectively a cylindrical closed structure all the way to the end of the tail till the bit shown by the arrow. This is a part of the neural tube that has not yet closed into a cylinder. It is still open. I'm going to zoom in on that bit of the tail, on the bit that has not yet closed in the next slide. But keep in mind how this arrow is pointing. It's pointing straight down onto the back of the embryo. There it is, on a slightly better microscope. So to orient you, this bit up top here is the tail end of the embryo, and the body will be down this way. And this structure is what the embryonic spinal cord looks like. It's got two parts, which I've separated out schematically here to help you visualize them. It's got a closed cylindrical-like section down at the bottom of the image, which is continuous with an open region shown in magenta here at the top. And this is one continuous structure which I've animated just to help you visualize it. It's quite a big structure relative to a cell. Each of these blue dots in this image is an individual cell. So this is a relatively big structure in a very, very, very small embryo. And what needs to happen during development is that this open portion needs to become a closed cylinder. If that doesn't happen, the embryo gets spina bifida. So that's the process we're interested in. How does this open region change its shape to form a closed cylinder? And we can get a bit of an idea of how that happens just by looking at the structure in embryos at different stages of development. I'm going to show you three in quick succession. Early, middle, late. So these three open portions of the neural tube at the tail end of the mouse embryo, all oriented in the same way, so tail tip at the top, body down at the bottom of the screen, are about 12 hours apart in development. And you can see that closure has progressed. This open structure, the structure which is still susceptible to forming spina bifida, has closed progressively over time. And as these arrows indicate, it's gotten shorter. And that raises the obvious question: how does the embryo manage to reduce the length of this structure? Well, to answer that question, one of the easiest ways to do it is to see if we can actually see this happening. But we can't do that in vivo, in a mouse, because the embryo in all mammals is going to be buried away in the abdomen, in the uterus. But luckily, there's a microsurgical technique we can use in which we can dissect out these embryos under a microscope, keeping them fully alive. These are at stages long before the brain and sensory systems have developed, and we can observe them growing under the microscope. So here we've got two embryos. The thing beating in the middle is the embryonic heart. Again, it's outside the body. It doesn't quite look like a heart just yet, but it will change shape over the subsequent few days. So the heart is beating. These embryos are both very much still alive. They're still pumping blood through their blood vessels. Up top, we've got the head that leads into the body, which curls round into the tail. In this one, the tail pokes in behind the embryo. These brown plug-like structures at the top are the placenta. This is what attaches the embryo to the maternal nutrient supply. And the balloons around them, holding the waters in, is the yolk sac. So these embryos are still fully separated from the outside environment and still continue to develop, so that we can observe developmental biology in real time right under a microscope. And using that, we can begin to look at the process by which this open region shortens. To orient you in the very brief video I'm going to show you next, here is the open region, this time shown in blue, sorry. This white box is the region I'm going to show you in the very short clip. It's a stop motion animation of about 2 minutes. And you're going to see cells on one side of the open region make finger-like protrusions that reach across the midline and grab hold of their neighbors on the other end. They're a bit like notches on a zipper. And if you imagine zippering from this point in the direction of the white arrow, that's going to make this open region shorter. All right. There's the finger-like protrusions reaching across, grabbing hold of their neighbor, and dragging it across the middle. That's a single notch on the zipper happening over a couple of minutes. You can imagine over hours to days, one notch after the other, that whole structure is going to zipper down and become shorter, reducing the area which is still exposed to the outside. Now, I like this animation because as well as showing the zipper shortening the open part, it shows that before shortening, you have to narrow the zipper. If the ends of the zipper were stuck far apart, the zipper would get stuck. It wouldn't be able to keep shortening the open section. And we see exactly the same thing if you look at the closing mouse neural tube. So again, we're going back to our early and late neural tube images. And you can see the open region in magenta has gotten shorter, but it's also gotten narrower. So how does that happen? Well, to visualize that, we can go back to our live imaging using one of our very recently developed setups. To orient you before I play this stop motion animation, this is the open region. This is the bit which needs to close to avoid spina bifida happening. This is the closed region, so this bit is safe. The bit in cyan is safe. And what you're going to watch over a stop motion animation that lasts about 2 hours is the open region narrowing the direction of the arrows. I'll let that play once again. So wherever you've got changes in shape of structures, of tissues, you've somehow got to have mechanical forces being generated and applied to change that shape. A body at motion will stay at motion. A body at rest will stay at rest. You've got to somehow apply a force to it in order to make it move. And when we started doing this research, we did not know where those forces came from. One possibility was that tissues on the outside of the open region could expand towards the middle, bringing the sides of the open portion together. So if you imagine the spring expanding slowly during the course of this video and pushing this open part towards the middle, that was one possibility. You can imagine having these springs on either end. Another possibility was that the cells in the open portion itself could generate the mechanical forces they need to pull the tissues around them onto them. You can imagine putting a blanket onto you and hiding under it. That's another possibility. It's practically impossible to differentiate between these two just by looking at a structure moving. The pink spring could be doing all the work, or the white spring could be stretching the pink spring. Now, if we had real springs in the embryo, I could go in, cut one, and see what happens. So if the pink spring is expanding and I cut it, the bits I cut would still keep expanding. If the white spring is constricting, then you can imagine what would happen if I stop that happening. Now, unfortunately, these springs obviously don't exist, and what we can do is look at the cells themselves. And we can cut individual cells or individual clumps of cells. Now, these are so small that the very smallest of scalpels would be far too big. But what we can do is use a high-powered laser and zap them with the power of the sun in a billionth of a second in order to vaporize a very thin line of tissue, and from that, see whether cells and tissues were stretched or compressed. So this works a little bit like this everyday situation. Those buttons were under tension. As soon as they're gone, the shirt rip pulls apart. That's exactly what's happening with these cells in the open region of this neural tube. So these cells were not being pushed together from the outside. They are not being compressed, but rather they and their neighbors are all themselves generating tension and pulling the sides inwards. That's at the individual cell level, but we can apply the same laser cutting technology to the whole tissue. So again, here's the open region, here's the closed region, and I'm going to use that laser to cut the closed portion. Oh, there it is. And having done that, we can analyze how tissues move when we stop individual portions bearing mechanical load. So what is the deformation of the tissue when we destroy load-bearing structures in it? And in this case, when we close the most recently fused portion, the portion that would normally be zippering down the length, that would be narrowing and zippering, what we see is that the tissue around it is pulled outwards. So if we had that pink spring expanding from the outwards inwards, that would hold the tissue in place. But instead, what we see is all this tissue on the outside pulls the open region out. And as that happens, the blue spring actually pulls that blanket over it a little bit more. It narrows the open region a tiny bit more. And it's from this sort of experiment and many, many more like it that we can point to the white spring in this animation and say, that's the one doing most of the work. That is the one which is pulling the blanket, pulling the surrounding green tissues onto it. I didn't tell you this, but the green tissue, the green cells in this image is what would go on to form the skin. So by covering itself in future skin, the open region of the neural tube is effectively contributing to that closure process in which the spinal cord will end up being below and covered by the skin. And over time, that combination of narrowing and reduction in length through zippering normally closes the open part of the neural tube. So this example right at the far end has almost completely achieved closure. This is the shape change that happens in the neural tube to build the embryonic spinal cord. But it doesn't always go as planned. There are a large number of genetic and environmental teratogens that will stop this gradual reduction in the size of the open region and instead result in progressive widening and enlargement. So these embryos at the bottom would not achieve closure of the neural tube, and they would get spina bifida. In each of these examples, there's a closed bit right at the top, and these areas that I'm highlighting with the pointer are where the spina bifida lesion would be. But this raises a very perplexing question. If you think back to that lamb I showed you in the beginning had spina bifida right at the end of its spinal cord. So why is it that this process of closure that starts off way up here and works its way all the way down the body. Why on earth would it fail when closure is almost complete? Why would it fail so catastrophically when it's almost done the job and completely covered the spinal cord? And we don't really know the answer to that question, but we've got a bit of a clue recently. Let's look at the shape of these open portions of the neural tube. Early, middle, late, practically closed. If you look at the shape of these structures, you'll see that it changes quite a lot. Early on, the open region is a shovel-shaped, spade-like structure that's wide open and has a relatively blunt ending end. Whereas at late stages, it's a much neater, elliptical structure. And in fact, as this transition happens, you may also notice that the far end, the tail end of the embryo, seems to move inwards towards outwards, away from the open region. So the most tailwards part of the open region ends up being within the embryo rather than reaching all the way to the end of the embryo. And that's a transition we've done quite a lot of work on recently. We've shown that at late stages, it seems to even form finger-like projections that appear to zipper towards the head end, towards the body of the embryo. So just as early on you had these zippering fingers that reached from the head down towards the tail end, we seem to have the opposite happening from the tail towards the head. So we could end up in a situation at very late stages of neural tube closure, the stages that would form the bottom of the spine, where you could have a double zipper. What we do know is that that tail end of the open part of the neural tube is itself a physical load-bearing structure. Now, biologists are perfectly accustomed to testing what genes do by knocking them out. So if you want to find out what a gene does, you knock it out and you see what happens without it. In mechanobiology, in studying the mechanics of biology, we do effectively the same thing using our lasers. So we can knock out or ablate that caudal part of the open region and see what happens to the overall tissue structure. And the fact that as soon as we cut that piece of tissue with a laser, the whole open region widens and becomes more spade-like, tells us this distal portion, this tail tip region, was holding the open section in a narrower, more readily closable configuration. So the tail end of the embryo at late stages of closure is a physical load-bearing structure that holds the open region together and potentially zippers up towards the body as well. Those are two characteristics that tell us it is initiating closure of the neural tube. It is a neural tube closure point. Now, there are a few closure points we know about. I didn't tell you this, but there's one which forms right between the brain and spinal cord. That is the very first closure initiating point. It zippers up partly towards the head and zippers down most of the length of the spine. In mice, there are two other closure points that form to close the head. And there was a fourth closure point hypothesized to exist. So if you look in the literature way back decades ago, there was the hypothesis that there is a closure 4 which forms right around here. We now know that closure 4 doesn't actually exist, but it is in fact the joining of zippering from closure 1 and closure 2. So these two closure points come together at what was hypothesized to be closure 4. Now, we've shown a new closure point at the tail tip. This is probably my biggest regret in my career so far. I have had to call this closure point closure 5. It is a misnomer because there is no closure 4. So future generations of undergrads and medical students and postdocs and PhD students are going to be trawling through the literature trying to find closure 4. And they'll think I can't count to 5. But so be it. There was nothing we could do about that. Closure 4 has been described. And we do now think that this terminal end of the neural tube, if it doesn't form correctly because of genetic mutations, of which we have one mouse model, or because of teratogens, including some medications, if that closure point doesn't form, then the result is spina bifida in the distal spine. So we think this new finding may start to explain why spina bifida is so common in the distal spine. And with that, I'd like to leave you with some take-home messages. First of all, that spina bifida is caused by failure of closure of this embryonic structure, the embryonic neural tube, during very early stages of embryo development. That building this structure is a complex process that requires carefully coordinated behaviors of cells in the open region around itself that effectively pull the future skin onto them and zipper down the length of the open part. I've told you folic acid supplementation is able to reduce the risk of this happening at a population level, but that it does not prevent all cases of spina bifida. Which brings me on to our need to understand why 200 people are born every year in the UK with this condition, why many of these are resistant to folic acid, in order to identify novel preventative strategies. And with that, I need to thank a large number of people, including Andy, who's been a fantastic mentor, and the Wellcome Trust, who've now funded me for 10 years in everything from bone biology to embryology. And I look forward to answering your questions.
Thank you very much, Gabriel. That was a tremendous lecture, beautiful images, and I hope you now know a little bit more about neural tube closure and spina bifida. We have time for questions. I would ask you, when you ask a question, to wait until the microphone reaches you, because this is being recorded, and if you don't speak into the microphone, the recording won't hear your question. So there's a question at the back.
Thank you. Great presentation. How is folic acid linked to the mechanisms of closing?
Yes. That's one of the issues with folic acid. We don't really know. There are many mechanisms that have been suggested, for example, by changing cell proliferation. But that in itself is a topic of ongoing research.
Also very impressed with the graphics and the nice presentation, even for a non-medical person. You mentioned at the end that there are 4, but they're called 5 closures in the embryo. I'm wondering, in an organism as complicated as a human being, sometimes you see reference to the sort of topological situation that you have, one opening in your mouth and one opening at the bottom of your stomach, but no others. Yet internally there are closures which are sort of splitting the topology of the animal into a number of regions. I mean, are we looking at just the beginning of hundreds of these things, or are there just 5 or 4 or whatever?
It's a very good question. And actually, the mouse seems to be more complicated than humans in this case, because humans don't have closure too. So if anything, at least in that situation in the head, we can drop one of them. Are we seeing the beginnings of hundreds of closure points? The very simple fact is it's very difficult to study human embryos. But the reason these were initially hypothesized to exist and then subsequently confirmed in a range of mammals is because of the distribution of different neural tube birth defects. So there are defects in which the entire spinal cord is open. That's where closure 1 has failed. There are defects where the head remains open, the brain remains open, called exencephaly. And that's a failure, realistically, of these 2 different closure points in humans that would meet at the top. Closure 5 had been hypothesized to exist in humans, although never identified. But we don't think it's likely that there are closure 6 and 7 intervening between closure 1 and 5. That seems extremely unlikely based on what we do know.
Question there?
Thanks very much for a lovely talk. I was just wondering, when does final closure of the tail tip occur compared to differentiation in response to sonic hedgehog signaling?
Oh, right. Okay, very, very scientific question. So you're going to have differentiation in response to sonic hedgehog signaling happening within the closed region even before completion of closure. But that's a program which is initiated and then continues throughout even much later stages of development in order to eventually form the motor neurons and the sensory neurons and so on. So the sonic hedgehog gradient will be established throughout this closed region, and you'll have the beginnings of that differentiation program already happening.
Thank you.
Just there, please.
Thank you very much. Can you give us an idea of what the incidence of spina bifida was before the use of folic acid?
So I can go to a hidden slide, which indirectly answers that by telling you the reduction in the incidence of spina bifida. In this case, in Argentina, which implemented mandatory folic acid fortification, and we are therefore able to compare the prevalence of spina bifida and associated defects before versus after folic acid in order to work out the percentage reduction. If you look at spina bifida that affects the thoracolumbar region, so quite high spina bifida, that in Argentina dropped by about 65%, and that's similar in Chile and so on. Whereas lumbosacral spina bifida, which we think is the type of spina bifida more likely to be related to closure 5, dropped by a slightly lower percentage. Yeah. So the answer is it depends on the exact type of defect.
Thank you. You mentioned that folic acid wasn't effective in all cases, that it's still prevalent. I'm sure you must be investigating as to why, but is this leading to a point where spinal bifida or neural tube defects will become a thing of the past eventually?
I mean, don't get me wrong, I'd love to put the surgeons out of a job, so I'd very much hope to. There are some very clear-cut situations in which individuals can have mutations in the pathway that normally responds to folic acid, normally metabolizes folic acid. So in that case, it doesn't matter what they receive, the end metabolite is not formed. But we don't know in many cases why individuals develop spina bifida, despite the fact that their parents still had folic acid, their mother still took folic acid in early stages of development. As we move towards personalized medicine and whole genome sequencing, we might get a bit more of an idea on a case-by-case basis, but we're not there yet.
Any more questions? Please.
The fact that folic acid is not so effective as it might be, does that indicate that there are other chemical products which may be more effective?
We'd certainly like to think so. So do you want to talk about the inositol study, Andy? Yes. Okay. So one of the sort of success stories, which is still in development, but it's looking promising, is by understanding the pathways involved, a different molecule called inositol has been identified, which in mouse models that are non-responsive to folic acid, reduces the risk of spina bifida. And that's entered a very small-scale clinical trial here in the UK, which was too small to reach a definitive conclusion. But let's just say it's promising. And there's a much larger clinical trial with this molecule being planned in the pipeline, led in large part by Andy and a colleague of ours, Professor Nick Green.
Could I ask you about the surgery you mentioned right at the beginning, the surgery in the womb, because that's in the media a lot at the moment, and it's just started here at UCL. 3 or 4 cases have been done in the last year. What effect do you think that's going to have on the prevalence, the frequency of this condition in the future?
It's a very difficult question to answer. So the main benefit of this surgery is actually not to the recovery of motor function in the limbs, for example, although there is some benefit there. The main benefit seems to be that by closing off the spinal cord, it allows a normal production of cerebrospinal fluid, the fluid that bathes our brain and central nervous system, allows it to be produced normally, and therefore secondary changes that commonly happen in the brain, in the hindbrain of patients with spina bifida become less likely with the surgery. Now, there is a genetic component to spina bifida. I wonder if this is the point you're aiming towards, Andy. We don't really understand the genetics of spina bifida. It's certainly very complex. But individuals who have one pregnancy affected with the condition are more likely to have a subsequent pregnancy, much more likely to have a subsequent pregnancy affected with spina bifida. What is not very clearly understood yet is whether someone who themselves has spina bifida is more likely to have a child with spina bifida, and whether there's going to be actually a greater need for these preventative strategies in at-risk populations. Question up there.
Your presentation was really good. You said that sometimes the brain's exposed. Do you know how many cases there have been of that?
Ooh. How many? So collectively, failure of neural tube closure affects 1 in 1,000 pregnancies on average. About 60% of those are in the spinal region in the period of folic acid fortification.
But open brain is actually very common because most of the other 40% are the open brain.
Yeah.
It's just that when you have an open brain as a fetus, you will not survive beyond birth. It's not possible. And so those babies are stillborn or die earlier in pregnancy, whereas those with spina bifida, although they have handicap and disability, they can survive until they're adults. Any more questions? If not, I think we should wrap up now. Let's thank Gabriel once again for an excellent lecture.